Okay, in this work, we are interested in the development difference between the brain and the brain organoids. The brain organoids are 3D culturing technologies which cultures stem cells to develop into the brain models. However, people are still unsure what is the difference and what's the similarity between the in vivo brain development versus the in vitro or brain organoid models. In order to answer this question, we have developed a pipeline called BOMA, which stands for Brain and Organoid Manifold Alignment. In this pipeline, given two RNA-seq datasets, one is for brain and another is for organoid. We first use a technology called global alignment, which roughly align these two RNA-seq datasets across the time. And after we get the global alignment result, we use manifold alignment to refine the global alignment and to embed all the samples onto the same manifold space. After we get all the embeddings, we then can infer trajectories, which stands for the pseudo-timing development, and we can also do cell clusterings, from which we can identify differential expressed genes, and we can do downstream analysis to identify the gene ontology functions of those genes, etc. We applied BOMA on a bunch of publicly available datasets, and here I'm showing you two examples. One is on bulk RNA-seq dataset, and the other is on single cell datasets. Within the bulk dataset, we aligned brain samples to organoids which cultured for about 2 years. As you can see, after the alignment, the brain samples and the organoid samples are in general well aligned, especially at the early time point. However, for the late stages, there seems to be some difference between the development between the brain and the organoid. We also aligned different regions of the brain with the organoid, which was summarized in this heat map. As you can see that the cortical region of the brain are in general better aligned with the organoids than the non-cortical regions, which is expected. However, within the cortical region itself, the different subregions also show some difference when aligning to the organoids, especially at early time points, which is earlier than the 100 days. So this indicates that the organoids are mimicking the brain development not unique, not the same across all the different subregions. We then applied BOMA onto single-cell RNA-seq datasets, which we integrated from multiple publications. As you can see that after the alignment, the different cell types of the brains and the organoids are aligned roughly at the same position. For example, the purple one, which stands for the exciter neuron, are aligned to the bottom right for both brain and the organoid. Then for the exciter neuron, we inferred the pseudo-timing trajectory, and for both of the brain and organoids, we then correlate this pseudo-timing with the actual timing from experiment time, and we found that a good correlation between the pseudo-timing with the experiment time for both within the brain and also within the organoid, which indicates our inferred pseudo-timing makes sense. We then identified the differential expression genes for both organoid and for the brain, we found that organoid genes were aligned to some specific trajectories of pathways, for example, the cell stress pathways. And for brains, the differential exchanges were aligned to some brain-specific development pathways, for example, the brain-derived neurotrophic factors, significance, signals, etc. We experimentally validated some 3 of the genes that we identified of which were differentially expressed between the organoid and the brains. So we have provided the community with a BOMA online app, which for general comparison between the RSIC datasets. We have also reported or submitted this work to bioRxiv with this preprint link and also the QR code. Thank you.